IHI, and I would like to welcome you to the first public webinar for a completed IMI project. And please note that more will follow for IMI and IHI projects. So the aim of these public webinars is to showcase the project achievements, boost the uptake of the project results, and demonstrate how the project has addressed key challenges in health research and innovation. So this is a great opportunity for the consortia to further disseminate their achievements and also to share their experience and the benefits of participating in a private-public partnership. So the project we will be discussing today is called iConsensus. So iConsensus is an IMI2 project which has started in 2018 and has been completed last year in 2022. Next slide, please. So more specifically, we're going to discuss about how to make a more efficient, cost-effective, and user-friendly process for the development and manufacture of biopharmaceuticals. Next slide, please. I am delighted to be joined today by 6 distinguished speakers. So first is Professor Véronique Choteau, coordinator of the iConsensus project and associate professor of industrial biotechnology at KTH. Dr. Luc Kuipers, who is the project leader of iConsensus for the technical part. And he was during the lifetime of iConsensus, he had the role of Manufacturing Sciences and Technology Fellow Innovation at Sanofi, which is the industry partner. And then Mr. Fabien Abeille, who is the co-coordinator of iConsensus and leader of the work package on system automation and integration and data monitoring and management, who is a senior R&D scientist at Micronit, which is an SME partner of the project. Also, Professor Asa Emmer, who is the leader of iConsensus work package on capillary electrophoresis chip platform, and who is professor in analytical chemistry at KTH. Professor Aman Rassom, who is leader of the iConsensus work package on affinity-based microfluidics and holographic methods, and who is professor of nanobiotechnology at KTH. And last but not least, Dr. Mark Dietrich Voss, who is project leader of iConsensus for the management part and is Director Global Alliance Management Public-Private Partnerships at Sanofi. So next slide, please. This is our agenda for today. After this short introduction, we will start with a discussion with the representative of iConsensus, and we will focus in 3 key aspects, which is the challenges that have been addressed by these projects and the objectives of these projects, the tangible results and their impact they achieved, and the exploitation of results and sustainability of assets developed. So after this discussion, we will open a Q&A session where you will be able to interact and ask questions to our speakers in writing. And I will explain in the next slide how you can do this. And finally, we will be concluding this webinar with some conclusion remarks. So before we start, let's make some housekeeping matters. So you are welcome to ask questions during the webinar or at the end of the webinar using the chat functionality, which can be found at the bottom of your screen. Please know that this session is being recorded, and the recordings together with the slides of the presentations will be made available on the IHI's website and on YouTube channel. Next slide, please. So let's start with this conversation with the iConsensus representative. And the next slide, please. We will start with the challenges and the objectives. So what is interesting here to understand is what was the challenge or the issue that your project sought to resolve, and what was the overall goal of your project, and why a public-private partnership was needed. The floor is yours.
Next slide, please. So first a little bit the explanation. So it's about the production of biopharmaceuticals, proteins, and there's a difference between classic pharma small molecule, like an aspirin, for instance, where you can see that that's a chemical reaction that is used to produce this aspirin. However, for biopharmaceutical or protein, it's far more complex. It's a chain of amino acids, of plenty of amino acids. And it is produced for this, it cannot be produced by a chemical reaction. So we have to use a living organism to produce the protein. Next slide, please. So biopharmaceuticals or proteins are different. They have large molecular weights. So a monoclonal antibody has a molecular weight of about 160,000. They are structural, very complex. They are heterogeneous. So that means that you are talking about a family of proteins. They also have impurities coming from the organisms that is producing the proteins. They are sensitive to physical conditions, so they are far more sensitive than the active substance in an aspirin. You have to use different kinds of assay to see the activity of the protein. For that, you use master cell banks. I explain that in a minute. And then a very important part is that it has post-translational modification. That means after a cell produces a protein, a chain of amino acids, there are some changes to the protein. And one of the major changes to the protein is the glycosylation. So on the right bottom, you see some sugar groups, and those sugar groups, complex sugars, they are connected to the backbone of amino acids. In the picture, you see those are blue. So the blue spheres are the glycosylation of a protein. Why are they so important? They are important for, first of all, for the correct folding, so the three-dimensional structure of the protein, but also for its activity, for binding to certain receptors, for after immunizing or after injecting into patients the immunogenicity, the half-time of a protein after injection, so what is called the pharmacokinetics, and the stability of the protein. So glycosylation is the one, is a very important post-translational modifications. There are others, but I'm just taking this because it's the most important one. And this is something that you cannot do chemically. For instance, the attachment of those sugar groups to the backbone of amino acids, you can do that on a very, very small scale, but not on a large scale. And if you want to use them as a therapeutic, of course, you need larger volumes. So that is why we use cells to produce those proteins for us. And the cells that are used are Chinese hamster ovary cells, are cells derived from a Chinese hamster originating from about, I think, 70 years ago, and still the most used animal cell line used in the production of biopharmaceuticals. Next slide, please. So here you see a small diagram with what happens. So we have a cell bank. That means that's a collection of tubes with all identical cells. Those cells are manipulated, so they contain a foreign gene that is of course there for the protein that needs to be produced. So you grow them up in a limited amount. You divide them over several hundred tubes. You put them into the liquid nitrogen, and you can keep them. So every time we take a vial from such a cell bank, we grow in several steps. We grow them until we have enough cells to inoculate a large bioreactor. On the right, you see this large bioreactor. That's a bioreactor of about 4,000 liters. It can go up to 20,000 liters. And that is where you grow the cells. So you grow the cells produce, secrete the protein into the nutrient solution, because of course you have to feed the cells so that they can grow. So you have, at the end, you get rid of the cells. That you can do by filtration, or you can do by other ways, by centrifugation. And then you have the supernatant, and the supernatant contains the protein. But of course, the cells themselves, and as I mentioned, most cell that is used is the hamster cell, are hamster proteins. And of course, you have to purify the protein of interest from this liquid. And that happens through several chromatography columns. At the end, you end up with a few 100 liters with pure protein. So that means that's protein that's more than 99% pure, so pure enough to be injected into patient. What you do is you formulate it, that you mean you add some substances to the protein, and then you divide that solution over tubes, those over flasks or vials, and that is the product that is then going to the patient to treat the patient. So that's in a nutshell a little bit where it's all about. But the important part for the iConsensus project is the cell culture production bioreactor, because cells are not difficult, but cells are not for the fully 100% controllable. And it's important that we always produce a consistent product at the end. And that depends, of course, on the cells, because the cells produce the product. Next slide, please. So as was said, the project started in 2018. For us, it started much earlier. I was at the time chairing ACTIP, which is a European organization, which is called the Advanced Cell Technology Industrial Platform. And this organization is around 30 years old, and it was set up about 30 years ago to really as a request from the EU at the time, because they saw that a new technology was coming up, and that was the technology, the animal cell technology. And so they wanted to have some people from the industry to look at this and to see a little bit what the potential could mean of this, of these animal cells. Next slide, please. So in 2012, so there always have been this connection to the EU. We had some EU representatives on some of the meetings that we had. And in 2012, we drafted a position paper which was talking about manufacturing, what we need to manufacture animal cells. And of course, by manufacturing animal cells, produce proteins. And that was the ultimate goal. Next slide, please. So we also visited together with Professor Hans-Jörg Hauser, who was at the time chairman of ESECT. So ESECT is the academic, so to say, animal cell culture organization. And we visited some people at the EU to see how we could work together. And they directed us towards the IMI2 program, and to contact people at FPA. And finally, we managed this, and we could get on the strategic research agenda for biomedical research. Next slide, please. So what you see in green are the sentences that we added. So there was a draft agenda, and so we said, okay, there needs to be something in there about manufacturing, because that is our core business. And so we could manage this, and we could, because it's not only for biopharmaceutical, but vaccines are sometimes produced by mammalians or animal cells. There are cell-based therapies, there are the ATMPs, all this which is related to animal cell use or human cell use. And so we succeeded in having this on the agenda. And that meant that there was an opening for us to really say, okay, now we have to look on subjects on manufacturing to get a project. Next slide, please. So why this public-private cooperation? Of course, you had the industry where we use GMP processes, good manufacturing processes, but the development of technology is not our core business. Our core business is growing cells, and cells produce the protein and then purifying the protein, put it into vials, and then bring it to the patient. On the other side, you have the academia and the SMEs, and so they are the one where a lot of innovation is coming from, and they could focus on new technologies. And that is where we brought this. In this project, we brought all of these together. So that finally ended up in the project, which then was called iConsensus. Next slide, please. So, and this is for me the last slide. So we within the active organization, we had Sanofi, Rentschler, Biontech, Bayer, GSK, UCB, Pfizer, who were interested in doing something. And the subject, so we had 3 subjects, but the subject that ended up into this project is we wanted to develop new innovative analytical tools to characterize cell culture fluid. So I mentioned that during production, cell culture is very important, that we need as much control of the cells as possible. And for that, we thought that there were not enough tools readily available. And so we said, okay, that is where that the SMEs and the academics could come in and look at this and give us the innovative tools that we finally need to be in better control of our cell culture. And this ended up in a 10th call for proposals. And of course, it's always in the IMI, it's an in-kind budget of the industry, and the same budget for the from the EU is then directed to the academic, so the other consortium consisting of SMEs and academics. So the budget for this project, the in-kind budget paid by those seven industry companies, was about 4.7 million euros. That means that the total budget for the project would be then 9.4 million euros. Next slide, please. So there were 10 expressions of interest, and of those 10 expressions of interest, there was one finally selected. It was selected by the industry partners, but also by the IMI scientific committee, and that was the iConsensus project, which was led or written, the consortium was led or written by Veronique, who will tell you more about that. And there you see some other companies and universities, SMEs, who were involved in the consortium. The next slide. Mark, I give the word to you now.
Mark, the floor is yours.
Thank you, Nick. Apologies, we have some technical issues, so you might not see me, but hopefully hear me. In the course of another year, in the end, it was about integrating and bringing together the public consortium, iConsensus, that formed itself after the call, and the industry partners. And in the end, to submit to the formerly known IMI office the full proposal, the stage 2 proposal, which then again was evaluated by independent experts and really integrating industry into the work package and having a team, a combined collaborative team. In the course of that, the multi-beneficiary model grant agreement was signed in 2018. And on the next slide, I think we had some mention as well on the so-called consortium agreement, which accompanies the grant agreement and is all about how the project is managed and how the governance is set up. And as you can see here, it's really truly collaborative. So every committee and all the work packages have an academic and an industry co-lead to really ensure integration of industry partners and SMEs in the most beneficial way in those consortia. Next slide, please. And I think that we are already through with part 1. In the end, it was May 2018, where iConsensus started with the first plenum meeting at the KPH in Stockholm. And I think I'm glad to hand over to Nick Arthur, the academic leaders, to present the outcome and impact of iConsensus.
Thank you very much, Mark. I hope that the people can manage to hear you. We had some technical issues because Mark could not join through as the other speakers have joined, but hopefully you managed to listen to what Mark has said. So thank you very much both for your presentation. And actually, it is interesting that you presented in a nutshell the idea generation process of IMI and actually how the history behind the generation of this topic that finally generated the iConsensus project. So now, next slide, please. I propose to move with the second part of this discussion, of this presentation. So let's talk about the results that this project has generated and also the impact they achieved. So the floor is yours.
Thank you, Salome. And if I can have the next slide. So here you recognize the slide of, look, I just wanted to make the connection. And on the next slide, you can also see the symbol of a bioreactor that we will use in the next slide. And next slide now. So here you have a representation a little bit alike what Luc has discussed, and here with the example of antibodies or monoclonal antibodies that we call this class of molecule, more generally biopharmaceuticals. So in a culture, you have a mixing of many different things. To start with, that the medium where the cells are grown include 70 to 90 different components, and then the cells are even producing themselves a lot of other components. And you recognize also to the right the correct quality that Luc has mentioned. He has spoken about the glycosylation. You have the aggregation possibly of these molecules, which is really damageable for even the safety. So these kind of features are really important. And in the next slide, you have also the symbolization of the fact that not only you want to obtain a high yield, but you also need absolutely to have the correct quality. And what is in the culture is really affecting or influencing the yield that you can obtain. So the amount of biopharmaceutical that you can produce, and at the same time, it also influences the quality. Another aspect, besides the fact that you want to produce these with these features of high yield and correct quality, you even want to decrease the process development costs. And this is also an aspect that we have brought together in the iConsensus. Next slide. So here is a high-level description of what we have in the iConsensus project. So we have focused on tools for online monitoring, for modeling, and for process development. And on the top left, you have the miniaturized analyzers, and I will let my colleagues Fabien, Osa, and Aman go more in deep in these. But if you look at top corner to the right, you have the online probes. So we've been looking at probes based on different physical spectroscopy like Raman, near infrared, 2D fluorescence, and these probes can be immersed directly in the bioreactor. These are technologies which already exist that we wanted to explore and to potentially combine. We've been looking, as the corner down to the left illustrates, we've been looking at high throughput microbioreactors for this field. This is really tools for process development. And then in the right corner to the down right corner, you have the mathematical models. And next slide. So one idea is that you might want to have models where you gather all the data that you can get from the measurement, and you provide a better understanding, but also tool that you can then use for optimization. And here is a representation of feedback control, which is based on some parameters, but you can include potentially all the different tools that we are generating in iConsensus potentially to also do this feedback control. And you can even include the information which is given by the mathematical models in your system. In the next slide. And here you have, again, the same representation of the technologies, but here illustrated in the different work packages. So you can see that on the top left, you have the miniaturized analyzers where we have had 3 different work packages and the interactions of several experts. So Cantisto, expert in capillary electrophoresis, Presense, expert in optical sensing, Micronet for the microfluidics, Paya for system based on multi-well for high throughput that we have been looking into application towards microfluidics, KTH for the more novel approach of capillary electrophoresis and affinity-based systems, ePresence for holographic imaging for the quantification of the cell concentration, ePratec for the for the integration of the software and hardware, and RAMCON for the online sampling device. Then if we look at the microbioreactor, we have had one work package looking into this from an SME M2P lab, which actually was later during the project acquired by the company Beckman Coulter. And also in collaboration with Aachen University. When it comes to the online probes, we had one work package looking at that, mainly directed by University of Hohenheim. And down to the right, you have the mathematical modeling by University of Mons and also KTH. And importantly was also in the center, the interaction with the EFPA members. And I would like to illustrate that with the continuation of the presentation of how we interact with them. Next slide, please. So here you can see the achievements that we have had. So we have had peer review publications. So this is a way which is really nice for showing the results and having also something which is staying. The SMEs have generated new products, and also new technologies have been also developed, and they will be developed also by my colleagues. We have been present at international conferences in presentations, and we have also had application notes, which are also important for the SMEs to show the possible application of what is done. The project has also generated 3 patent applications. And what is really nice in this type of consortium is the possibility to have new collaborations and new exchange. And for instance, the SMEs, they have new ideas, they have prototypes, and suddenly they have really a really good platform for trying, for beta testing the new product or new application. Next slide. And here you have some example of applications in industry. So to the left, you have the company Presence. So they have developed new sensors in a way that they have been able to also test these at industrial partners. They have now sensors which are autoclavable. They have also sensors which can be installed on a shaft so that they can be immersed in a bioreactor like conventional sensors instead of being only dot sensors like they usually have. In the corner to the right, you have the system of the SME ePresence. They have integrated the holographic-based cell density quantification system in a commercial high throughput system. And then another example is the realization of the SME PAIA, where they have achieved a new type of high throughput system for detection of the aggregation. Next slide. And so these have been the applications which are already available, but here on the present slide, you can see the ones that we would like to push further. So where we have seen a proof of concept for the miniaturizer sensors, but we will need a little bit more time for achieving the commercialization, because these ones have been really new, and the goal with the project was the proof of concept, and we knew that we wouldn't have really products with these, so this will need still quite some time after now the proof of concept which has been generated. Next slide. And if we are thinking about reflection about how the public-private partnership has been helping, I think it was really clear for the SMEs and for the academia which were in the consortium, that the fact that we had the interaction with the EFPA partner really gave us the industrial relevance of what was done. So that means really concrete application. And one aspect is that if you speak with industrial partners, they know exactly what they would like to see in terms of kind of results. And of course, we're coming with idea of products. So it's not so that it is done in one day, but they can tell you the limit of detection they want to have, the stability. They can project to what will be the needs for GMP. And this is really helpful because otherwise this is something which is absolutely too abstract or where there's no knowledge potentially in academia and some knowledge in SME. So these are the industrial constraints, but also the regulatory aspects are really important, because if you are doing from the beginning something which will not hold for the regulatory aspects, you will hear from industrial partners that there's no point even to start. It needs to be at a point possible to be compliant for GMP if you are wanting to implement that in GMP at a time. Of course, you also have direct contact for trying the prototype in industrial environment, where they have also access to an equipment which is much better and much larger, and this is quite important. And another aspect from outside of these more regulatory aspects or industrial constraint, it's also the real motivation and the enthusiasm that we have seen in the team. So the people in industry, they have themselves kind of background knowledge about other things, and they are kind of enthusiastic about seeing potential new applications and potential new ways for them to do things. So there's really kind of a mutual enthusiasm for the progress to come. And I think now I will give the word to my colleague. Next slide, please. Yeah, now I think we should deep dive a little bit in the miniaturized sensors, first with Fabien.
Yes, thank you, Veronique. Okay, I think there's some images on the left side that don't really appear, but anyway, so here just another picture kind of summarizing the project iConsensus and trying to see where this work package shouldn't fit. So as we mentioned before, it's important that we can monitor the quality of the product that are made within the bioreactor. So usually these rely on traditional lab equipment, but those ones are usually very bulky. So if you need to an entire room basically to characterize a lot of different things, and you can't also have like a real-time feedback on the culture using this equipment. So the idea of one of the idea in the projects, the iConsensus project, was to develop this AIAS, so at-line integrated analytical system, which has two main components. One is this miniaturized module that Veronique mentioned earlier, and my colleagues will deep dive a bit more in some of them later on. So trying to bring to use miniaturization so that we can integrate them on a single system, and then at the same time we want to be able to control those modules, but also interact with maybe the online probes that was Veronique mentioned earlier that are attached to the bioreactor, and also then feed data to the mathematical model. And this was then fit into the platform Paola that we call, so the platform for online analysis. So if we go to the next slide, and we look at what we've effectively built in terms of integration with this miniaturized system. So the idea was to be modular, so that, as Veronique mentioned, we could then have some of the modules push further into commercialization if they seem to be working very well and relevant, but also be able to have a system and also be flexible, because a lot of things were still developed, tweaked during the project. So the idea was to use a liquid handler, custom liquid handler that you see on the top left corner. So this has the size of about a tabletop, and bring on to use those miniaturized modules. And you see on the right side the deck of this liquid handler, where we can find the affinity module, the gap electrolysis module, and also this optical sensing module. And with that, there's a lot of other slots where we have heating and cooling block. This is if we want to do some formulations to help some reaction, we want to heat. The cooling part is more to stop reactions or to host some reagent for a longer time. And then also a bunch of labware like filters, pipe tips, and other type of tubes and vials to basically do a lot of different operations. So when we use this analytical module, usually upfront, we need to, once we take a sample from the bioreactor, we need to prepare the sample or some reagents so that they are fresh and they can be introduced in this analytical module. So this is what the liquid handler can do in collaboration then with those modules that are on board. If we go to the next slide. And then, as I mentioned, the second component was to be able to collect data, also interact with those modules, and also provide some interface for an operator to use the system. So this has been also strong work and development there to develop a server so that we can have also then people that can be registered as users, expert users, or like engineers, operators, for instance, with different levels of ways to tweak basically recipes that you want to the system to perform, and control the different modules through some other hardware that is also embedded there, and also visualize all those data that are made real time. Next slide. So in the end, what we've managed to build through this Work Package 7 is this integrated platform that hosts 3 analytical modules that can perform about 8 analyses at once. There are actually some potential more that can be performed. These were still developed during the project and still need to be strengthened, but we could even go up to 12 of those. And this takes about less than a milliliter of sample. And you can complete about 1 to 3 full, like those 8 analyses can be performed 1 to 3 times per day, basically. So this, when we heard from the FPI partners, this was actually a good achievement, because this really gives a lot more information for the state of your culture and try to basically correct how the culture is going, qualify it, and get the best yield and quality out of that. So with that, so we gave like the general architecture of this alias system, and now we leave the floor to my colleagues to explain a bit more some of the analytical modules.
So next slide, please. So I'm Åsa Emmer, and I was the lead of VP3, as Luc or Veronique said earlier. The objective of this work package was to analyze some of the components in the culture media and also some aspects of the pharmaceutical product. So I will go through some why, what, and hows of this work package. So starting with why, this has also been touched upon by both Luc and Veronique, because the rationale by doing this analysis is, of course, to get the correct product and in the end get the safety for the patients and the product or a pharmaceutical that is correct and gives the right result. And at the same time, manage a good, efficient manufacturing. So in the end, the cost for patients and society will be decreased. And as always, we also need to have our sustainability glasses on to make as little waste as possible, use as little chemicals as possible. So what we wanted to see was that enough, but not too much nutrients was put into the bioreactor to keep the cells viable and efficiently producing, in the end, the pharmaceutical. Next slide, please. So what were we looking for? Nutrients, products, and byproducts. So Luke mentioned that these biopharmaceuticals are really complex molecules built by amino acids. And to be able to build them, of course, you need to have the right amount and the right type of amino acids. So that needs to be checked. Then we also looked for sugars and vitamins, and that is because the cells needs an energy source, and they also need both sugars and vitamins to grow and be viable. And in the end, the correct molecule without aggregates or fragmented was important because you want to have a good monoclonal antibody as your final biopharmaceutical. Next slide, please. So how did we do this? Of course, since this is a very complex mixture, the cell media, we needed to separate our analyze the components, the substances that we were looking for from all of the rest material in the media. So we choose capillary electrophoresis, CE, and also chip CE, because you need very low sample volumes to perform these kind of techniques, and you also consume very low amount of chemicals and produce little waste, and at the same time you get fast analysis, and the footprint of this instrumentation is very low. So the main principle of this type of technique is that you separate your analytes depending on their charge, and also a little bit by the shape and size, but mainly the charge. So next slide, please. So in iConsensus, we used capillary electrophoresis, the traditional version, in thin capillaries with a length of 33 centimeters. First, to develop all the methods, because it's easier to do it in this traditional mode first. And then we transferred some of the methods, not all of them, but some of the methods also to the chip setup, where we only had 8 centimeters. So on the right-hand side here, you can see an example showing the separation of some sugars, both in reference samples and also a spiked media sample and real media samples. And as you can see, the result is rather similar between the traditional capillary CE and the microchip CE, but in the microchip CE, the total analysis time is half or even shorter than in the traditional one. Next slide, please. So was this work package successful or not? Yes, I would say it was, because we had in the end a very nice CE chip model. You have seen it in both Veronique's and Fabienne's photos. We developed methods for amino acids, sugars, vitamins, and the product. And we developed CE chip methods for amino acids and sugars. And there are standard operational procedures documents for all of these methods. So anyone that has these SOPs can perform the same analysis. These methods were also used for different types of production samples, and very useful. It was also tested by 3 of the industrial partners in their settings with the same kind of setup, but with their own real samples. So thank you Beyondis, Rentschler, and Sanofi to do this. So in the end, I want to show a short video on how the chip setup, or actually only the chip separation, works. So please start the video. So you can see here a great, oops, it went very fast. You should see in the top right corner a blue cross. And if you start it again, you might see the injection, some green stuff coming. No? Well, then I can explain what you should have seen, and that is, yeah, now you can see it. The green stuff coming, that's the sample. injected in the injection channel, and then the sample, the green sample, is injected in the separation channel, the blue one. And then you can see the actual separation of these purple, yellow, and red components. And at the bottom, you will get your electropherogram with all the different substances at each peak. So thank you. Next presenter, next slide.
Thank you, Åsa. And my name is Aman Rusam from KTH. I'm going to describe the affinity module that we've been developing, more specifically microfluidic-based affinity module. So what you're looking at here is you would take the supernatant sample, put it into this affinity-based microfluidic system for, on one hand, looking at the efficiency of the manufacturing process, and on the other hand, on the safety and efficacy of the product. So if you look into the efficiency, we have developed a multiplex microfluidic-based system to look at the product as well as impurities in parallel. And for the safety and efficacy, this we do by monitoring the glycosylation profile. So in the next slide, I will show you how it looks like. As Fabienne mentioned, this is the prototype. We have on the left side the hardware reader looking for the chip, and if you zoom out in the chip, I show you the schematics of the 4 parallel channels filled with beads. Each channel looks at one target. In this case, we're looking into the host cell protein, the target protein, LDH, and a control. And on the right-hand movies, you see the lower part, how the beads are filled into the channels. So each of the channels are filled with unique beads looking for one type of target, while the sample then comes in from the, in this case, lower left, and go out into the upper, lower right into the upper left, and then passing each of the chambers for interaction with the targets. So for the results, I will show you in the next slide how the microfluidic affinity is performing. So in the blue squares are conventional gold standard methods, and the red and black are the results from the microfluidic systems. So all in all, the correlation is quite nicely using traditional ELISA methods when you compare it to ours. So if you look at the left-hand side is the host cell proteins. The time interval is basically for the different days, and they follow hand in hand. In the right side is the antibody production. Also similarly here, singleplex and multiplex means that a single channel or multiple channel, they are following the same trend. Maybe something to highlight is that for the LDH, we've developed a competitive assay, meaning that if you do not have an LDH, our signal will be high. And if you have signal LDH, it will be lower. So all in all, for the efficiency, it follows nicely with conventional methods. And for the glycon assays, in the next slide, we can show. So what we've done is that we're taking the SME PioBio from Germany. They have developed a microtiterplate method using lectins and fluorescence method. We've taken that assay and adapted it to microfluidics by adding it to the beads and using a colorimetric method. So in the left bar graph, what we're looking at is 4 different harvest days. So H6 means harvesting at day 6, day 12, 21, and 26. And as you can see, the microfluidic system follows nicely conventional methods. In this case, the conventional methods is based on HPLC mass spec, while here using this immunoassay, we could easily differentiate between high signals from low signals. So these are very quickly the summary of the microfluidic system in this work package. I thank you and leave it back to Veronique.
Thank you all for the presentations. Before we move to the third and final part, I have a couple of questions for you. So you have nicely presented the numerous tools developed per work package, but how about the bigger picture? So now that the project has ended and you have an overview of what has been achieved, would you describe this project as successful?
Yeah, I could answer. I think it has been quite successful in a way that we had this view of developing new technologies, and we have been successful in really doing this. Of course, we had a view which was really long and almost like a product, and we have no product for the miniaturized sensors that we have showed here more in detail, but there have been many other technologies in parallel which have been actually commercialized and with new applications. So I think that the balance is really positive, and of course we would like to push further now the new technologies that you have seen develop a little bit more in detail. But really a success story, I think, for our part.
Thank you, Veronique. I agree with you. So I have another question. An important achievement of, it's basically more of a comment plus a question. An important achievement of this project has been the collaboration between academia, so with the experts providing know-how, with the SMEs providing innovative technology and expertise, but also the interaction and engagement with the end users, which is the industry. So I have a question for the industry. You had the possibility to test the technology produced in your industrial environment. So how beneficial was this to basically offer a proof of concept of what has been developed in a lab scale? And did you have the possibility to provide comments in order to further adapt the tools developed to better fit your industry needs?
So if I may talk for the industry, this, of course, yeah, this collaboration is always very fruitful because there you end up having new ideas. But as I mentioned, we are not into the technology development. But what we saw is that we have prototypes and you can use them as modules, so you can use them separately. At the end, you can combine them in what Fabien mentioned, the Elias system. It's prototype. So that means that there still needs to be some development work in bringing this really, that it could be commercially available and that the industry could say, okay, I now put this next to the bioreactor and I do my measurements. So that is a little bit still, there's a lot of potency in there, there's a lot of potential in there that we at the end succeed in doing that, but it still needs some further development to make it, so to say, user-friendly, because the original idea was that it should be operated by manufacturing people on the floor, so you don't need a PhD to do all this, and that was the goal, but that's where we are not there. But the industry is definitely very interested in this because we are still lacking a little bit what we can measure in the cell culture fluid and be in better control of the cells. But this is definitely something, if we could then develop this a little bit further to a user-friendly commercial system, which you can just put in module or the total, the Elias system, would be very, very helpful. But we are very happy also. It's not only that you want to have something, but you develop also a lot of knowledge, and a lot of knowledge which is then be used in other ways within when you are growing your cells.
Okay, thank you, Luke. And I also have a question about the impact on SMEs. And Fabien, this is for you. So several of the prototypes were developed by SMEs partners. We've seen some of them. And some of the products are already commercialized and some require further development for commercialization. So what was your experience as an SME in participating in this IMI project?
Yeah, so there's different levels of let's say return of investment, if we can say. I think the first thing which is really important is that this collaboration we have with the industry. So because in our case, they will be our customers. So we have a direct, let's say, link to them, and we can listen to their needs and understand what they care most about when we try to develop our own technology. So that's the first part, and then I think there are two aspects. So some of the SMEs managed to really develop new products, but for some, even if new products were not there, it's also a way to strengthen their technology, and this is something we have done as Micronists through the products, trying to improve on some of our technologies. So they were not mentioned here, but for instance, we have some integrated pneumatic valves that we can use into microfluidic devices and other type of technologies. So it's a very nice, let's say, place to get more, let's say, through those products.
Okay, thank you, Fabien. So let's slowly start, let's move to the third and final part of this conversation, which is on the exploitation of results and sustainability of assets developed. So what has been put in place in terms of exploitation and sustainability? And are your results publicly available? The floor is yours.
Thank you, Salome. So then I will continue. Yeah, thank you. So in terms of exploitation of the results, so we have already touched upon the fact that we have developed, or the SMEs have new products which are available, and they were also depicted in the part which was results. A lot of new knowledge, which is difficult to really measure in some way, but that has been a really nice learning. And not to mention the fact that the SMEs, they already know what their market is and what the needs are. They want to have these more precise kind of information from the industrial partners to really align with the needs. But when it comes to the academia partners, they are expert in what they really know well, and we have example here of Aman and Osa. They are really expert in the analytics that they have shown, but they do not really have a feeling for what the industry is really desiring, really needing. So this is really kind of new knowledge, and now they can embark to develop new types of products with the knowledge. They have a better understanding of the needs, and that's kind of, this type of knowledge is really important. And then new applications, and we have also discussed them, and some of application notes are out there and they are accessible. And of course, in terms of what is sustainability is also the peer review publications that are available. And so there's in the next slide, you see a slide that I will not go through, but this information is available both in the IHI site and also in our website of iConsensus, but this lists kind of the different links to the peer review and to the application notes for all the different technologies. And then in the next slide, this is also another question which was asked. which have been the new collaboration and funding opportunities and the visibility. So when it comes to the visibility, one really important thing that I think I have already mentioned several times is the fact that when we are working with the EFPA partners, that gives us the relevance and the view from the industry. So then when we are speaking with other people, they have this knowledge that we already know what industry wants. So really the relevance is important. And also when you are speaking with other people outside from iConsensus, if you have the fact that you have been interacting and collaborating with these large industrial partners, that really gives you kind of a label of quality, which is really important for speaking then with others. So that also gives the possibility to have other funding for other projects, which can be parallel or associated, etc. And also, I mean, generally speaking, I think people are quite well aware of the fact that to be part of the EU project is always a kind of a label of quality and achievement, and certainly also this type of partnership with IHI. And then in the next slide, the question was also about what are the progress which are brought to the field, and I think we have already touched upon that. Potentially with the miniaturized instruments that we could apply for monitoring processes, we can get easiness of operation because these systems are simpler in comparison with sophisticated instruments that can really reduce the cost of goods, so the cost for these analysis, and this is easier to implement in terms of automation. And basically the simplicity is also a positive factor, which will potentially come or provide robustness, etc. Because at the end of the day, of course, we want to achieve a better control of the process. That gives a higher patient safety in itself from more information which is delivered in the process in order to make a correct decision, to improve the robustness, the reproducibility. But also we can of course use these analysis for process development. So suddenly we have the possibility to do more measurements. So we are in a field which is also going towards more high throughput development, but where certainly the analytics is the bottleneck in order to support the high throughput, which is possible for the culture. So we need to have solutions also of high throughput for the analytics. And the fact that we could use this miniaturized system is also quite positive. And of course, if we can improve the information that we can give to the process development, that means also that we can at the end have better process and achieve faster solutions, which is always a benefit for the patient also. And then in the next slide, you have even a vision that Luc has started to discuss as well, which is the application even for not only to the left to monoclonal antibody, which has been the subject of iConsensus, or glycoproteins or enzymes, but also for these new therapies like cell therapy and gene therapy. So these are really less mature than cultures like antibody production, and we really need an arsenal of new ways to monitor and control these processes, which are today not robust enough, not reproducible enough, and which really need also for which the yields need to be increased and the costs decreased. So to bring more automation, more monitoring can certainly help also this field. And this is really something that should be actively continued. And then I think I'm coming to the last slide, which is the next slide, where also Salome was asking to summarize shortly iConsensus. And as you can see in this sentence, it was not possible to have it shortly, but we think that the fact that it was an ambitious project has been really described both by the reviewers when we started the project and also by ourselves. But we are proud of the results that we have obtained. It has included new products, new application, new thinking technology. And we have really allied our different brains from academia, from SMEs, and from industry to really put all this knowledge together. Thank you.
Thank you very much, Veronique, and thank you, everyone. It was a really interesting presentation / discussion. I would also like to congratulate you for this great project iConsensus that I have had the honor to be your scientific officer since the beginning. Now I would like to ask the audience if they have questions for the speakers. So you can use the chat at the bottom of your screen to ask questions in written, and and we'll give you a couple of minutes. So in case there are questions, you can send them in writing. In the meantime, if we can change slide, please. Yeah, next slide. So you can stay informed about IHI news, calls, and events like the one you've seen today. They will follow more through our website. You can in LinkedIn, and you can follow us on Twitter. For the moment, I don't see any question. I also wanted to ask you about, in terms of TRLs, so the TRLs, the technology readiness level, which is a way to measure the maturity of the technologies generated. Have you estimated the level reached per technology developed? And have you put an objective of what you would like to reach as TRL in the future?
So in one of the last deliverables, we have systematically listed the TRL achieved by the different technologies, because there are many, and honestly I don't have them on top of my head by heart, but we have done these kind of what was the objective from the beginning, because that was described in the proposal, and what we have achieved. So we have progressed quite some on many of the points that we wanted to achieve. It's difficult to give one TRL for the whole project. There are many different
of course, but
technologies.
It's a measurable mean to have a starting point and where you have reached and where you would like to go further. So I can see a question. How do you think your learnings and outputs could contribute to the transition towards a more sustainable, a greener biomanufacturing?
I think that in terms of being more green, there's one tendency nowadays in the field, which is to go towards continuous bioprocessing. And continuous bioprocessing has been described as being greener, and indeed it is an intensification which really reduces the size of the bioreactors and allows also to use systems which have much less energy needs and clean water and CO2 footprint, etc. But they also need to have more of this monitoring that we have developed. So this is where I think we can, the type of technology that we are developing for online or inline measurement can help this field. And generally speaking, in terms of failure of runs, maybe Luc can relate more to that, but on a general basis, sometimes industry has to throw away batches because they have not followed the profile which was wanted. And to have better control can be certainly something which at the end is also more sustainable. I don't know, Luc, do you want to comment on just that part?
Yeah, for the sustainable part, I would think then if you look at what the industry is moving towards is that they are going to having much more control during the process itself. And so now what they sometimes do at the end of the process, they are looking for the characteristics of the produced protein, and then they find out that it's not according to the criteria, and they have to stop everything. So they have to say, they have to do an investigation what was wrong, but also they have to throw away, because if the product is not consistent, it cannot be used in the patient. And even small differences sometimes can mean that a product is not used. So in that way, I think sustainability is guaranteed by having more control of your cell culture and doing the type of online at-line measurements helps with that. That at the end you're going to those and you mentioned, so you have those continuous processing, and the idea is that when at the end of the process you can approve your product because you have tested during the production so many parameters, at-line or in-line, of your process that you can say, okay, my process behaved quite well. It behaved according to our criteria within the ranges that we want, so that you have the proof that it is already a consistent project that is produced. And that is a little bit where we're going to, so that makes it much faster in production yeah, better in control, immediate release after production. So it would really make more runs per time possible as well. So it will increase a little bit. So that is the sustainability. The greener part, of course, depends on what type of culture you are using. There are new ways of culturing, but what we used in the past is also stainless steel. So I think at the moment that wouldn't change that much, but it's more the sustainability, the rapid processing, the be better in control to produce this consistent and safe product for the patient.
Thank you. And I will take this opportunity now that we are talking about greener biomanufacturing to communicate on our IHI call, topic 6, which is on sustainable circular development and manufacturing of healthcare products and their quantitative environmental impact assessment. That is somehow linked to this discussion and is currently open for applications. I can see another question. Is there further protection in terms of patent applications planned, or are other companies actively encouraged to adopt some of the developed technologies, and so patent protection is not sought for that reason?
We have already some patent applications for some of the applications of iConsensus. We have not patented some of the technologies because they were prior art, but there it's like we usually do. I think in the continuation that we would like to do, there will potentially be other opportunities for patenting, but at this stage, we have protected the technology that was new without prior art.
Okay, thank you. And now I can see that my colleague has put in the chat the link for the call for environment topics, which are online, and we have the link here. So we are slowly reaching the end. I cannot see any more questions. I would like to thank you one more time for this great conversation that we had, and that I had the pleasure to start with you these new public webinars for our completed project. So that's it from me. I don't know if you would like to say something.
I think we would like from all the consensus, the consensus, I should say, we would like to thank you, because you have been kind of supporting us all the way. That has been quite a nice journey. I think also that has been a journey with really nice colleagues and new friends. So that has been quite nice journey to be in one of these projects. A lot of work, but nice work as well, and kind of synergy and joy of doing things together is always nice.
Yeah. I agree with Veronique according to this, and I always want to mention that we are always available if there's more information needed on certain of these topics. So just contact Veronique or somebody else from the team, so that at the end we could even bring this to a further level as we have at the moment. Yeah, thank you.
Mm-hmm.
Yes, exactly. We would like to come to a commercialization.
Yeah.
Definitely.
Super. Thank you. And before ending, I would also like to promote a little bit our platform on collecting ideas for helping us making new topics for calls for proposals. So it also, everybody can propose ideas at IHI through our website. And also if you feel that you have, you believe that further research is needed in a specific topic, in a specific area, don't hesitate to submit your ideas. And these ideas will pass through a certain process, and we can use part of it or to make new topics for our calls. And that's it. Let's close for today. Thank you very much, and all the best.